Welcome everyone to our Quercus Quest campaign training webinar. This webinar is part of our Phenology Week celebration, so I hope that you've been joining us for the other webinars this week, and we'll have some more information later about some of the other events happening. My name is Erin Postumus. I'm the Partnerships and Projects Manager for the USA National Phenology Network. I'm joined today by three of the researchers who are part of this larger effort that is behind the Quercus Quest campaign. So I'm going to be passing it over to them in a couple minutes for them to share some of the research that they're working on and why the phenology data that you are collecting as part of this campaign is so important. So we have Rebecca Mon, who's a postdoctoral researcher with the Morton Arboretum. We have Laura Ostrowski, who is a doctoral student at the University of Minnesota. And we have Ian Pierce, who is a research ecologist with the US Geological Survey, joining us today. So to give you an idea of what we're going to be going over, we'll first start out with an overview from our research team. Then I'll be sharing some background on the USA National Phenology Network and Nature's Notebook. And then I'll share some background information on how you can get started with being an observer as part of Quercus Quest. We'll share some training materials, and then we'll make sure we have some time for Q&A. We are recording this webinar. We're going to be sending out the recording to everyone who registered. You'll see that at the bottom of your screen, you have a Q&A. I invite you to put any questions that you have for the research team or for the NPN staff in that Q&A. And we can kind of get to that during the webinar and make sure we get to any unanswered questions at the end as well. You can also post in the chat, and I welcome you now if you'd like to tell us where you're calling in from. You can also let us know any signs of spring that you're seeing where you are. Today is officially the spring equinox, so happy spring if spring happens to be happening where you are. But feel free to put something in the chat if you like to tell us where you're calling in from. At this point, I'm going to stop sharing, and I'm going to be passing it over to Rebecca to kick us off.
Hello, thank you all for coming, and I'm excited to share about how we're utilizing your research. The team of researchers is a national and international group, so there's 6 institutions represented as part of this research group that's looking at different aspects of the data. And then we're also collaborating with international researchers in order to think about how what we understand here in North America applies to our global understanding of climate and phenology. My research is specifically looking at the Eastern North American white oaks. This includes 16 good species, and many of these species co-occur. Here I've shown maps of 7 of the species. They've been pulled out into different maps so we can see them all, but you'll notice that a lot of them overlap in their ranges and have slightly different leaf morphotypes and slightly different occurrence ranges. One of the interesting things is that a lot of these species hybridize. So here I've given an example of a Quercus macrocarpa or bur oak and Quercus alba or white oak that's hybridized to produce offspring. These offspring often are able to produce viable offspring themselves, and so we can have genes passed down into these hybrids and then sometimes back into the print. Is fairly common, and there's a long history of gene flow between these species. Previous hybrids between all of the 7 species I showed you previously are known to co-occur, and we've found evidence for a number of these in our wild data looking at their genomics, which I'm showing below. The ones that we have data for are the ones with the thicker lines. And so this suggests that there not only is hybridization, but then there's also gene flow where genes are moving between species. And this is likely key to the ecological and evolutionary success of these oaks. When we think about these oaks, we're not just thinking about the trees themselves, but also the associated community. So we have A, which is that tree that includes the phenology that the physiology that we see in H, as well as the genetics that would include the hybridization that underlie this species. We also have the leaf endophytic fungi that live in the leaves. We have the gall wasps that parasitize the leaves and branches. We have the endomycorrhizal fungi and ectomycorrhizal fungi that grow in and on the roots, as well as the whole community of co-occurring plants, whether that is other oaks or non-oak species that they co-occur with. This research is really looking at all these different aspects. We're looking at the oak evolutionary history, the genetic diversity within oaks, the hybridization between the different oaks, the oak functional traits, the communities of organisms that live on these oaks, and to try to understand how these different aspects, their history, their hybridization, their functional traits, and the community, how they all influence each other. And so far, we found that our genetics do support that hybridization is occurring between some of these species, but we also see that the hybridization is localized. So some populations have lots of hybrids, others don't have as many. And your work collecting the phenology data can help us understand if this variation in hybridization is due to the variation in phenology across the range of these species. So you might imagine that some places they might flower at very different times, and other places they might flower at a similar time. And so that's how we hope to utilize your data to tease that apart. One previous study that's looked at this has to do with the live oaks or the section varieties. And they looked at two different kinds of oaks, Quercus geminata, or the sand live oak that lives on drier soil, and then the Quercus virginiana, the southern live oak that grows on moister soil. What they found is that the flowering time is actually fairly different between these two species. And they most of the time don't hybridize, but occasionally they do. And so we're hoping to do something similar, looking at the phenology data of Quercus macrocarpa and the co-occurring trees. So by collecting that phenology data, both the flowering data and the leaf out information of bur oak and then the co-occurring white oak species, you're helping us understand the species potential to cross-pollinate. So thank you so much for collecting that data, and I'll pass it on to Laura.
Okay, thanks, Rebecca. I'll just share my screen. 1 second. Okay, Erin, are you seeing that?
Yep, we can see it.
Okay, perfect. All right, well, thanks so much for having me. I'm excited to be here today talking with you. And some of the research that I do is in this Dimensions of Biodiversity project that Rebecca explained. And much of this work really centers around questions about climate change and about how trees can adapt to new environmental conditions, changing temperatures and changing precipitation. And one way to get at this kind of question experimentally is with a common garden experiment, where we plant trees from populations across the geographic range of a species in a common environment, and then test experimentally for local adaptation and for plasticity. So it's really a way to understand how trees can respond and survive in different environmental conditions. And for the work that I do, I focus just on one species of oak. This is bur oak, Quercus macrocarpa, that I'm sure many of you know. It looks like that picture on the right. You might guess from the name macrocarpa that it has large seeds, very, very large acorns. And it has a huge geographic range. You'll find it all the way down in Texas and all the way up to Canada. And so you can imagine that populations of bur oak throughout its range experience very different climates. And so even though in that area, it's generally all the same species. Some of it does hybridize, as Rebecca said. But although it's the same species, the populations are experiencing different climates and may have local adaptation to the different climates that they're living in. And so I'm part of this exciting experiment called ACE, which is an acronym, Adaptation to Climate and Environment. It's a series of common gardens in a reciprocal transplant experiment. So we have gardens in Minnesota at Cedar Creek, in Illinois at the Morton Arboretum, and in Oklahoma at the Kessler Field Station. And our research team collected bur oak acorns in all three of those areas and then planted them out reciprocally in those three gardens. So for example, you'll have bur oaks with acorns sourced from Minnesota planted down in Oklahoma and vice versa. So we can see how a population adapted to a northern area may actually fare in warmer and drier conditions like Oklahoma. So there are lots of different people working on this, many people. It's an exciting, large project, and I couldn't possibly share all of the results with you, but I'm just going to show three quick results that I'm excited about, some of the data coming out of this reciprocal transplant. First, this is some mortality data. So this is survival broken down by the gardens and populations. So what you're seeing here is the three panels along the top are the three different gardens, and the colors are the populations. So orange is seeds sourced from Oklahoma, the Oklahoma population planted out in these three gardens. And what we're seeing so far, a few years into this reciprocal transplant experiment, is that there is a lot of mortality in the Oklahoma garden, especially these Illinois and Minnesota and more northern populations in green and blue are experiencing a lot of mortality when planted down in these hotter, drier conditions, which may be somewhat foreshadowing how those populations might fare in hotter and drier climates. Another thing we're looking at is leaf morphology. There are many different ways of quantifying the shape of a leaf and lots of traits that we're interested in. One that I'm particularly interested in is this trait called circularity that basically tells you how loby an oak leaf is. So if you look at the two pictures of oak leaves on the bottom here, the one on the left is very dissected. The sinuses go in very far toward the midvein, so that would have a low circularity score on this estimate. Circularity, if the score was a 1, it would be a perfect circle. So no leaf will have a 1, but a lower score is a more dissected leaf. A higher score will be a leaf that looks more like the one on the right, less dissected with very shallow lobes. And what we're seeing so far with the data is that the Oklahoma population tends to have this more loby form. And actually that lobiness or low circularity correlates with fitness in these gardens. So on the y-axis here, you're seeing an estimate of fitness. We use Astor models to estimate the fitness of the individuals in the gardens. And we're finding that the leaves that are lobier, more dissected, or low circularity score actually have higher fitness, and the families that have lower survival, lower growth, so a lower fitness score, tend to be the ones that have a higher circularity leaf. So this is one example of many of the traits we're looking at, where we're finding that certain traits may be under selection and could be adaptive between these populations and could also be shifting plastically between different environments. And then lastly, I just wanted to show phenology. Of course, we've been collecting phenology data at all three of these gardens, mostly spring leaf out and fall senescence. So this is showing the fall senescence from this past fall in Minnesota and in Oklahoma. As you can see, the Oklahoma garden, those trees are senescing much later in the year, which makes sense. But what I'm excited about with this data is if you look at the orange line, the Oklahoma population within these gardens, that population is senescing later. We're seeing that the trees from Oklahoma have later leaf color change and later senescence, no matter which garden they're planted in. So that some of those phenophases may be traits that are under selection or traits that are genetically different between the populations, which is pretty cool. So we're also incorporating data from the National Phenology Network, data that I'm sure some of you all have collected from bur oaks in these three areas, and looking at bur oak data for Minnesota, Illinois, and Oklahoma to put this into context. So thank you so much for the work that you do on phenology. And there are many people involved in this work here, a few of them and a few of the funding sources I'd like to thank. So thank you so much, and I will pass it on to Ian.
All right, thank you. I'm going to just pull up a presentation really quick. Yeah, excellent. Can people hear me?
Yep, we can hear you.
Okay, excellent. Great. So yes, as people have talked about, we're really interested in the phenology of oaks. These are trees that are foundational species and have tremendously large ranges throughout North America and really throughout all of the Northern Hemisphere. So this map, again, is that map of macrocarpa that we've all seen. And you can imagine there's quite a lot of variation in the key phenological traits of this oak throughout that range. Some of that variation is just due to the environment, and that is the information that we can learn very well from the National Phenology Network data that you all are helping to collect. So we will be able to understand why oaks have different phenology in their more northerly range from southerly by fitting some sort of model that tells us about the weather events and things that drive the phenology of this tree. And we can, of course, stack that up with other oak species that co-occur in order to understand something about what we predict to be where these species would have overlapping phenologies and where they would not. We also have these trees planted outside of this range. So I've always been kind of curious just what is happening with oak species when they aren't in their range at all, when they're experiencing environments that are actually pretty different from where they evolved. We know some of this from these reciprocal transplant experiments, but if you happen to be in one of those areas outside of the range of bur oak or other white oak species, I'd personally be rather curious to know when are they beefing out and what is driving their patterns of phenology. So oaks, another characteristic of phenology of oaks, it's really kind of exciting, is that they seem to have a lot of variation even within populations in key phenological events. So this is a photograph that was taken up on a hill in California on the same day. These are two oak trees of the same species, in this case, valley oak, in California. And you can see that this tree on the right has already set flower and its catkins have senesced. It has a bunch of leaves on it. I'm not sure exactly what phenophase this would be at in the NPN framework, but it's pretty far along. Whereas this other tree, same species, really close together, is there on the left that has not yet started its spring. And this happens year after year after year with these two trees. So they probably have not made it, is my guess, even though they are very close together. So the reason for that study was that this seems to drive certain events in oaks. So some years you have trees leafing out all at about the same time. You have a fast spring, it warms up really fast, and all of the oaks of a species set flower more or less at the same time. Those tend to be the years where you have really good acorn crops, which would suggest that this process of masting in trees is at least in part driven by that phenological synchrony of when they're flowering. With the idea on those slow springs when trees do not flower all at the same time, that there might be pollen limitation, which limits the seed production of those trees. My main interest, though, in the phenology of oak trees has to do with all of those other creatures that live on them. And many of those creatures that live on them, particularly the insects, have phenologies that are very in tune with the tree. That there are quite a number of these insects, for example, that are actually out there preceding bud burst and sitting around waiting for the buds to break and the leaves to start to form, because they feed on the really early developing tissues of oaks. So many of these insects are their populations are probably driven in large part by the phenology of oak leaves. There was a really interesting study out of Missouri, one of my favorites, that showed that you get booms and busts of a lot of the spring moths on oak trees, which seems to correspond to whether their phenologies match with the bud break of the tree in that particular year. Whereas for the late season ones, where phenology maybe doesn't matter as much, they tend to have fairly even populations from year to year. The group of organisms that I'm most excited about with this and I'm studying in the context of this project are the gall wasps. And many of you have probably seen these, particularly if you spend time staring at the buds and branches of oaks. These are actually insects that coerce the plant into making these strange-looking little houses for them. So each of these little blips or nubs or thumbtack-looking things has a small wasp larva living inside of it. These aren't like stinging wasps like you might imagine coming unwanted to your barbecue, but are tiny little things that you'd probably think of as like a gnat or something like that. So they're innocuous creatures, and they create all of this really interesting diversity up in oak trees, where in a single oak tree we can have as many as 12 or 15 different gall wasp species. And those in turn are fed upon by a great variety of predatory insects and parasitic insects. So you have this little self-contained food web. Gall wasps have 2 generations per year, so they're very in tune both with the fall phenology of leaves, when the leaves are senesced, and also the spring bud break, when they're often parasitizing buds or those fast-growing leaf tissues. So with the data that you're collecting, we'll understand a lot more about the phenological variation in oak leaf out throughout the United States, and hopefully be able to understand the dynamics of these insects in a better way because of it. Thank you.
Thank you all so much for sharing all the different aspects of research as part of this project. It is a really interesting project because it does have so many different parts to it, and just a huge researcher team that's looking at lots of different dimensions. So it's great to hear that the phenology data is useful for so many different aspects, from understanding whether or not trees can hybridize with each other to understanding the connection between phenology and all the organisms that are using oak trees. So thanks again for sharing all that. Okay, so just going back to our agenda here, we're going to move into some more background information on Nature's Notebook, and then I will be talking to you about how you can get involved as part of Quercus Quest. But first off, just to kind of get a sense of the people that are joining us today, I'd love to hear a little more about how you all participate in keeping track of plant and animal life cycles. I have a poll that I'm going to launch that is all about how you track phenology, if you do. Maybe you're already a Nature's Notebook observer, maybe you collect phenology data in a different way, or maybe you're not yet participating. So I will give it a little bit for people to answer. We already have a bunch of responses coming in. We already have quite a few Nature's Notebook observers who are here, so thank you for being here. All right. So we have about half of the attendees are already Nature's Notebook observers, which is wonderful. Some people contribute to other online data collection platforms. A lot of people are nature photographers taking pictures of plants and animals, and a couple of people don't yet track plant and animal seasonal cycles. So the USA National Phenology Network was established back in 2007. At that time, there wasn't really a standardized way to collect information about phenology in the US. So we were really established for that purpose, to be a standardized way for people to collect data in the same way across the country, to really enable people to compare data across years, across locations, across species, and really facilitate that learning about phenology. So we continue to be, that is kind of our primary purpose, is to be the collectors of the data, as well as storing the data and sharing it. And all of the data that are collected are made freely available for anyone to use. So the primary way that the data are collected is through Nature's Notebook. This is the USA-NPN's Plant and Animal Observation Platform. This map shows locations of observers that have recorded data, and then the circles on there show the places where we have lots of data coming in. So you can see we have lots of people in urban areas, kind of along the coast, but we're always looking to fill in those gaps in places that we don't yet have people observing. We have to date collected over 35 million records as part of Nature's Notebook. So we're just so grateful to all of you that are participating with the program and helping us to collect this really important resource. We have a number of these data collection campaigns that Quercus Quest is one of. These are efforts that are usually initiated by researchers that have a need for a type of phenology data on particular species. Sometimes they're also started by natural resource managers who need data as well. So you'll see we have a variety of campaigns that are more at the national level, things like Green Wave, which is interested in tracking the leaf out of maples, oaks, and poplars, as well as the leaf color change in the fall. That's located all across the country. We also have some more specific campaigns like Flowers for Bats, which is in this part of the country in Tucson, Arizona, so southeastern Arizona and New Mexico, interested in the nectar availability for the migrating lesser long-nosed bats. We also have a Nectar Connectors campaign that's focused on nectar plants and the flowering of those nectar resources for pollinators, and that's also a national campaign. And then Quercus Quest, which is why we are gathered here today, is an effort that's mostly in the eastern US, although as Ian says, we're interested in these oak trees wherever they may be across the country. And so that's what we're gonna really get into talking about now. So this is the third year of Quercus Quest. And so just to give a glimpse into last year's data, we had 123 backyard observers taking part in this campaign, and including 46 local phenology programs. These are groups of individuals that come together. They're often led by organizations, nature centers, arboreta, university classes, and they come together with a shared purpose for collecting phenology data. And they often have a local phenology leader who engages with a number of volunteers or students. So our top contributing LPPs from last year included Meredith College, Earthwise Aware, the Morton Arboretum, Crosby Farm Park, and then the Mount Auburn Cemetery citizen scientists. So thank you all for your efforts in collecting data for this program. Just to give a snapshot of what these data can look like, this is white oak data collected by observers at Meredith College. And so what we're looking at is on this graph here, we have each of these lines represents basically a day that someone went out and made an observation in Nature's Notebook. The colored lines indicate someone said that they did see this life cycle stage, initial shoot or leaf growth occurring. And then the gray lines indicate that they looked, but they did not see that. And you can see that they were pretty consistent in their observations throughout the springtime. And then there's a gap here because the students weren't there for most of the summer, and then they started again in the fall. But I like this because it shows that even if you're only able to observe for a little bit of time, we can still collect some really useful information about what's going on in that springtime period where the oak trees are just starting to leaf out. This group was able to capture the initial growth as well as when the full-size leaves were on the tree, and then also the open flowers when those started and ended. We can also look at the data in more of a map format. This one shows the symbols on here represent a location that's collecting oak data. The shapes of the symbols indicate the species that was being collected there. And this map actually is only a subset of the observations that were collected last year. And the ones that are showing up here are ones that collected their first yes observation of open flowers with a preceding no within 7 days. And I'll get into more of that later and why that's important. But these are kind of the places where we have a more precise estimate of when open flowers first started among the observations collected last year, at least for these 3 species of white oak, bur oak, and chestnut oak. Another way we can look at last year's data is by looking at one of these curves. So this is showing over the course of last year, the proportion of people that were saying yes for leaves in a few different species. And so we can see that among most of the species, they generally started having leaves in about mid-March or so, with a peak starting around May. With the exception of live oak, which is not deciduous, and so it kept its leaves on throughout the year with a little bit of a dip, maybe when it had some seasonal leaves falling. And then we can also look at the flowering phase. And again, live oak shows it's quite different in its timing of when it has flowers in comparison to some of the other species where they're a little bit more consistent in when the peak amount of the yes reports for flowers was showing up in about mid-May or so. So moving on to how you can get started as part of this campaign, I'm going to go into these steps in more detail, but just to give an overview, basically it requires creating a Nature's Notebook account, adding a site, adding one of the oak species that's on our list, recording your data, and then making sure that you sign up for our campaign emails so that you can hear back from us about what we're finding with your data. So for the first step, you can get started by downloading the Nature's Notebook app. You can find it in the App Store or the Google Play Store. You can also do all of your observations online, but we found the app is pretty useful for being able to set everything up. So definitely recommend starting with that. Once you have your app and you have your account and you open up the app, you'll then create a site. This is called a personal site, which is distinguished from a program, a local phenology program site where you're actually observing with a group of people. So you click on that button to create a site. In terms of how your site should be set up or what encompasses a site, it's pretty flexible. If you're just observing a single oak tree, that can be your site if that's all that you're tracking. If you also want to track other things, maybe a couple of oak trees, maybe a few of different species, or maybe an oak tree and something else that's part of one of our other campaigns, or maybe you want to do an oak tree and a couple of animals, then you should try to figure out a site that makes sense in terms of the convenience to you, representative habitat. So you want to pick something that kind of represents the area around you. Uniform habitat if you can. If you're in an area like this diagram that has lots of different kind of ecosystem types, try to create a site that's fairly consistent. You can always create more than one site to kind of encompass the different ecosystems that might be near you. And then in terms of appropriate size, if it's just an oak tree, then your site is just the size of the area around your tree. If you are observing animals, then think about what's the area that you can reasonably kind of account for the animals that you're tracking. Are you in a meadow where you can see to the other side? Maybe that's the size of your site. But that convenience, I think, is the most important thing, because we want you to find a location that is going to be an area where you can go to frequently. Don't pick something that's in the perfect natural area that's miles away from you that you probably won't ever go to after the first time. Try to pick something that's convenient enough where you can get back to it with some consistency. In terms of the species that we're interested in, we're primarily interested in white oaks and bur oaks and their relatives, although we do have a couple of live oaks on the list as well. So this is the full list. This is on the Quercus Quest website if you need to check back on that. And so you can, once you pick out the oak species that you can identify near you that's on this list, you can add it to your site by clicking on that Add Plants and Animals button on the app. If you are already a Nature's Notebook observer, you're welcome to add oak to an existing site if your site kind of encompasses an area where your oak is, or you can add a new site. Maybe there's an oak tree down the street, but it's not the normal site that you use. You can set up a second site that includes that oak. This is an example of what the data sheet looks like for bur oak. So basically with Nature's Notebook, we try to make it pretty simple as we can. So we have a series of yes or no questions. That's really the basis of the data sheet. So we're asking you things like, do you see breaking leaf buds? And your options are yes, no, or this question mark, which means I'm not sure. And you can always use the question mark. You can change your data later if you figure out, oh, it really was a yes or it was a no. But I always recommend using that question mark if you're uncertain. You can also answer all or none or only a subset of these questions. So if you feel like you're not quite ready to answer the question about breaking leaf buds, you can always just skip that question if you're unsure. For this particular campaign, we're really interested in the leafing questions or phenophases. Phenophases are just the life cycle stages like these here. And we're also interested in the flowering phenophases as well. And you can always record information about the acorns on your oak trees too, or the leaf color in the fall. But if you feel like you're limited in time and you want to just do a subset of the questions, we recommend starting with these. We also have this full list of what we call phenophase definitions, and those are meant to help you and take any guesswork out of the observations that you're making. So if you're unsure what a breaking leaf bud is, make sure that you use the definition and read that through carefully. Those definitions are available on the app. There's a little eye icon next to each of the phenophases. You can always tap that if you need a refresher. That'll bring up the definition. And those are available on the website as well, and you can print them out in a guide form. For some of these particular phenophase definitions, we even have species-specific information. So you'll see for this one, we actually say what you can expect to see for that particular species. And we have that for like the fruit definitions as well to know what a ripe fruit will look like in comparison to an unripe fruit. So just to get into a little bit more detail about the leafing phenophases, I like this graphic because it shows that there is some overlap in between these different questions. And you don't have to just pick one of these to say yes to. Often you'll be saying yes to multiple of the different types of questions, the leaves, flowers, and fruits questions. So for example, when you first start observing your tree, say it just started having these tiny leaf buds, you'll start saying yes for breaking leaf buds, but you'll say no for the rest of these phases. Once you see that it's transitioned out of breaking leaf buds and you're starting to see some actual leaves, then you'll start saying yes for leaves, but you'll continue to say yes for breaking leaf buds as long as some of the leaves are still in that breaking bud stage. And so really what you should be doing is consider each of these questions as separate. So don't feel like because you said yes to one, you have to say no or yes to the other. Really consider them as separate things and answer them independently. For flowers, it's similar. So once you start saying yes for any sort of flowering structure, which is the flowers or flower buds, then eventually some of them will start to open and you'll say yes for open flowers. But there's still some sort of flower structure on the tree, so you'll still say yes to flowers or flower buds. So again, you're considering those as separate. You're answering, do I see any flowers or flower buds? Yes. And do you see open flowers? Yes. So you would be saying yes to those at the same time. And so to get into some more specifics and an example of what these flowers actually look like, oak flowers are kind of funny. They are in the form of what we call catkins. So they're flower structures on the tree. Often they can be kind of hard to see. These first two are the male flowers. So these are often, they can be at the ends of the branches. They might be a little bit more visible to you. There might be some closer to the ground. You can get up close and see them. This is when they first start appearing. They're kind of tightly clustered, and then they start to open up and expand, kind of like an accordion. The female flowers are really hard to see. You may never see them if you're looking at a large tree. They're often kind of tucked into a little, at the center of the stem here, very close to the branch, and they're very tiny. So just to kind of go through how you would record if you were looking at these in the field. For this photo, you would likely say yes for breaking leaf buds. This is just the leaf tip visible here. You don't see the full part of the leaf with the stalk. For increasing leaf size, you wouldn't say that yet because it's still in that breaking leaf bud stage. You're not really looking at a leaf. No leaves yet. For flowers or flower buds, yes, there's some sort of a flower structure here. But open flowers, no. These are still pretty tightly clustered together. So you would say a no for that one. In this central photo, you would still, because of this little thing here, say a yes for breaking leaf buds. This is where the leaf is actually expanded and you can see the leaf stalk. So you would say a yes for increasing leaf size as well as a yes for leaves. For flowers or flower buds, again, there's some sort of flower structure on here. You would say yes. And then now, because the flowers have expanded, they're considered open. So this would be a yes to all 5 of those. As for this last one, we can't see any breaking leaf buds in this picture, so it's kind of an unknown. You could say no for what we're seeing in this little picture. I just put a question mark because we can't even see where the leaves would be on this. And then also unknown for increasing leaf size and leaves. For flowers or flower buds, yes, there's some sort of a structure. And I said open flowers, no, because this does seem still very tightly clustered. Again, you probably won't see female flowers close enough to be able to really use them for your observations. But if you're looking at the male flowers, then you would be saying a yes for those phases. So I mentioned that you have a couple different options for data collection. We recommend using the Nature's Notebook app. You can set up your account there and make your observations there. But if you prefer, you can also get things set up on the computer on what's called your observation deck. And that's where you'll set up your sites and add your plants. And then you can also print out paper data sheets from there, which you can then enter online. And we have an interface that very closely matches the data sheet. So a little bit more on kind of why the data collection frequency is important. So if you'll remember the calendar that I showed earlier, the times that people go out to observe, each time you go out, you're saying either a no or a yes. And the more frequently you go out, the more precise we can kind of estimate when a phenophase first starts. So for example, if you're only going out every 6 days, there's this period of uncertainty, which is 6 days long, where we don't know where the flowering actually started. It was sometime in between April 3rd and April 9th. If you're going out every 3 days, then we know that it's only a period of 3 days of uncertainty, which is much better for the data quality. It makes it easier for us to kind of look at differences between years. We can start to look at relationships with climate variables like temperature, precipitation. So if you're trying to do the highest data quality, then try to do more frequent data collection when things are changing. So when you're looking at the first breaking leaf buds or the first time you start to see open flowers, and then you can observe less frequently when things are already going. Maybe the tree already has a lot of open flowers, it's leafed out. You can taper off and do less frequent data collection at that point. We also have a second part in addition to the yes or no questions, and that's what we call the intensity questions. So if you say yes for flowers or flower buds, you'll be asked a second question that's how many flowers or flower buds are present. And these are also optional. So if you feel like you can't do that yet, that's totally fine. Just skip that, just say not sure. But if you'd like to try to estimate intensity, you're really trying to put your estimate into one of these bins here. So we're not telling you to count exactly how many flower structures are on the tree. We're asking you to try to estimate, is it 101 to 1,000? Is it 1,001 to 10,000? And so on. And often you can pretty quickly rule out a lot of these intensity bins right away. And then you're often trying to figure out, oh, is it this one or is it this one? Is it less than 10,000 or is it more than 10,000? And we have some tools to help you with that estimation I'll talk about later in our observer certification course. And then another thing to keep in mind with this is really what we're trying to get is a peak in when the most structures were available on your plant. So don't worry so much about getting exactly right the bin. What we're really looking for is that progression over time and being able to see when was that peak and when was the most open flower, the percentage of open flowers, or when were the most breaking leaf buds on the tree. If you make 6 different observations spaced out by a week throughout the year, then you will earn your Quercus Quest badge, and you can see that on your observation deck. So something to look forward to after the end of the season. Okay, so now we have a couple different slides that we're going to do, and I'm going to launch a poll. So these polls actually they're meant to kind of be very similar to what you'd be looking at on the data sheet. So I'm going to launch one, and it's a series of these yes or no questions, yes, no, or question mark, for a number of phenophases. I think I have the leaves and the flowers phenophases on here. And so you'll see I put the full phenophase definition, so it might take you a while to kind of read through that definition. But once you get the hang of it and you've done this a few times, you'll kind of know what you're looking for, and it'll be much quicker to kind of go through these. But feel free to take a little bit of time and look through the definitions. If you scroll down, you'll see there's a number of these, I think 5 total questions on here about the leaves and the flowers. So, and I know it's very simple just to look at a single photo like this, but just for the sake of testing it out, just look at what's in the photo and then try to make your best guess. And of course, you're always welcome to use that question mark if you're not sure about what you're looking at. And I'll give it a little more time. I know that there's a lot to read through at first. In the meantime, I will check on the Q&A. Okay, we have about half of people have participated. I'll give it a little bit more time. Looks like we've had some good questions about gall wasps in the chat for the Q&A. Again, make sure you're answering the questions separately, kind of considering them each individually. Okay, I'm going to end the poll and let's look at the results. Okay, so for do you see breaking leaf buds? Again, we're looking at a leaf tip that's visible, but before the first leaf has unfolded to expose the leaf stalk or the petiole. So most people said yes, which I agree with. I think we could say this is the leaf tip. I know that it says green leaf tip, but we do have this species specific information at the end that for this species in particular, the leaf tips can look reddish. So you guys weren't fooled by that. And then that's most people said yes for that, which is great. As far as leaves, I would say no for that. We don't yet see that leaf stalk. We can't see the full leaf. So it's still just in that breaking leaf buds category. Oops, oh gosh, sorry about that. And then as far as the leaves, that's also, it's not a leaf yet. So that is also a no. And then for flowers or flower buds, and then open flowers, right, we don't see any flowering structures yet. So nice work. Okay. Here is the next one. I'm going to launch another poll, which should match the last one, except for maybe a little bit of species specific information about this particular one. But the questions, it's again, the 3 questions about the leaves and 2 questions about the flowers. So hopefully you're starting to get a little more familiar with the definitions. You've read them through once, so getting to know them a little bit and getting to know what you're looking for. If you, if you close the quiz accidentally, you might go to the more in the panel and see if you can relaunch it. I don't know if you click on that it will come back up, but it might be that it's gone for now and you can do the next one. Sorry about that. Okay, I think most people have participated. Okay, I'm gonna end it. So for this one, do you see breaking leaf buds? This one is kind of tricky. I feel like there might be some breaking leaf buds in here. It would be much easier if we were there and we could look at this a little bit closer and kind of go on different sides of it. So I would probably put a question mark for breaking leaf buds because I'm not sure with these. This one might be, but I can't really see if there's a leaf stalk there. As for the leaves, yes, I think we can definitely say there's leaves. These are fully unfolded. We can see the leaf stalk. We also, like, we can kind of guess, even though we haven't been watching this tree and seen, what it looks like with full leaf size. These look like they're small leaves, so we can say that they're not yet full size. They are still increasing. It gets much easier to make this estimation once you've seen your tree, what it looks like at full leaf out, and you can kind of compare and say, oh, I know that this is a leaf that's only, a 25% of the full leaf size. As for the flowers or flower buds, yes, there's definitely flower structures here, so that's a definite yes. As for open flowers, again, it would be easier if you've seen this tree, over the course of many weeks and you know what it looks like when the flowers are fully open. These are not open yet, I would say. I think if we looked at this in a couple weeks, we would see they're unfolded, kind of like that accordion opening up. And we would say that they're open flowers at that point. But again, much easier once you've been looking at your tree week after week and you know when they're starting to expand and become open. All right, I think we have one more. Okay, one more. And this one, I think I did not put, I don't think leaves because it was just kind of hard to see. But let me see, I think it just has 2 questions on this one. So this is just the flowering questions, since it's kind of hard to see the leaves here. Okay, so got about half of people, a little bit more. Okay, I'm gonna end this one. So this one, definitely there are flower structures here. So that's a yes for flowers or flower buds. And then open flowers, yes, this does look much more expanded. And you can even see some of the flower parts exposed. So that would be a yes to both for those. So nice job, everybody. Okay, so then I mentioned that make sure that you sign up for Quercus Quest messages. I'll have a QR code at the end where you can just do that right now. Or if you go to the Quercus Quest landing page, there's a sign up for that as well. So we'll put the links in the chat for those later. But it's definitely good to sign up for messages. You'll, get some prompts on making sure you start looking out for leaves or flowers. We'll also give you results of what you and others are reporting as part of the campaign, and then updates from the researchers as well about what's happening with the data that you're collecting. So a couple special considerations for oaks. It can be difficult to identify oaks, especially before they start leafing out. You can use the leaf shape and acorns once you have those available to look at. There's also this great field guide. This is also linked from the Quercus Quest landing page, so we'll get that link in the chat. Our researcher team might have some other tips or resources to help. iNaturalist is a great resource for species identification as well. Also keep in mind that some trees don't produce acorns every year, and some trees also will mass, so where they have a lot of acorns in a particular year. So don't be too concerned if you don't see acorns in a particular year. And also keep in mind that only the female flowers will produce the acorns. We're really interested in sites that have multiple species. So if you do have a couple of the oaks on the list, it would be wonderful if you could observe both of those species. And then we're also interested in particular site conditions. So whether your trees are getting any additional water, whether you fertilize in your yard, and whether you know the planting date. And so unfortunately, you can't add that information in the app right now, but you can do that on the website. So if you sign up on the app, you can use your same login on the USA-NPN.org website, and then navigate through the Observe with Nature's Notebook to your observation deck, this is what it looks like here. And then if you go to edit site, the site where you have your oak tree, we have some details here where you can indicate some information about the development in the area, whether your site's urban or rural, where the closest water is and that kind of thing. So you can fill out those details and then make sure you click the save button at the bottom of the page. And then as far as other details on the screen where you can look at your individual oak trees, if you go to this add or edit plants, you can add some more details about whether your plant is wild, if you know, whether it gets any sort of supplemental water or fertilizer, if you know the planting date, you can even indicate the shade status, and then you can put any other information you'd like in the comments, and then make sure you save that as well. I mentioned we have a lot of resources to help. So those phenophase definitions are available to print. We also have a great botany primer guide that has lots of photographs, so you can look at more information about catkins and things like that. All that's available and linked from your observation deck on the website. This is the observation deck in the learning tab. So here's the link to that botany primer. There's also a phenophase primer that is still kind of in development, almost ready. Some FAQs. And then the observation or the observer certification course is something that I definitely recommend taking. There are 5 modules. The first module is kind of an overview about some of the stuff I've talked about today, how to set up a site, how to, add a plant, things like that. There's one on the mobile app in particular. And then there's also one on estimating intensity. So that'll really help you if you're interested in doing the estimates of the number of flower structures or the percentage of open flowers. That'll walk you through how to do all of that as well. We also invite you to explore the data. A lot of those visualizations I showed are ones that you can make yourself. So our visualization tool, it's linked from the website and there's the direct link here, but that allows you to explore your own data and then data collected by everyone else. And then we also, because the data are made freely available, we have lots of researchers that download and use the data. And we try to do summaries of these publications at least every other month. So you can see those on our news page, and we also put them out in our newsletter. So you can kind of learn about how your data and others' data are being used for a lot of different aspects of research. I also wanted to mention our local phenology program. So you can observe as a backyard observer at a personal site, but you can also get together with a group of people and observe together. So you can contact our volunteer engagement coordinator, Samantha, if you'd like more information about that. We have lots of resources available, including a certification course for local phenology leaders. We also have monthly calls that we host. So we would invite you to join any of those. And then I think this is my last slide, but definitely join us for our other Phenology Week activities. We've got a local phenology leader Q&A on Thursday. We have a phenology wheel paint along on Friday, where you can make a phenology wheel based on phenology data, things that you like to see in different seasons. And then if you happen to be in Tucson, we're doing a phenology meetup at the Tucson Audubon Mason Center on Saturday. Okay, so this is my last slide, I think. So just to recap, to get started, the first thing to do is create a Nature's Notebook account. You'll want to add a site and at least one oak. Add some details if you can to tell us about the conditions at your site. Make sure you sign up for messages. Take advantage of the training materials that we have. And then ideally record observations once a week, but really anything that you can contribute is wonderful and we appreciate it. And feel free to reach out to me. I'll also put my email in the chat. And then here is the QR code for the Quercus Quest messages to sign up for those. It's also on the landing page, which we can get in the chat as well. But I know there's not a lot of time for questions, but hopefully we can take a check in on the Q&A and then get to any unanswered questions as well. Okay, so I'm going to check in on, looks like people have asked about sending out the chat text. Yeah, we'll when we send out the summary email with the webinar recording, we can definitely include some of the Q&A in that as well. Let's see, I'm just looking through some, I know a lot of questions have been answered by our researcher team. Thank you so much for that. Just looking at the other open questions. How do we take a tree off of our list if it's cut down? So going back to that add or edit plants page on your observation deck, you can delete a plant and then it'll ask you why you're deleting it, and you can indicate that it was removed or it died. Maybe you're not observing it anymore, or maybe you made a mistake and it's a tree that we don't have on the list or something like that. But you can indicate all that when you delete your plant. If you delete it because it was cut down or it died, it won't delete your data. It'll just remove the tree, and so you won't have to look at it anymore, but the data will stay in the database. I found that the submit save button in the app didn't always transmit. I would say, so on the review screen on the app, there's a bar that says either user data are up to date or user data are not up to date, and it's orange if it's not. That's when the data haven't made it into the database. So the only thing you can really do is tap that sync button on the review screen, and that should work to get your data uploaded. Make sure you're on a good Wi-Fi connection. If you're not on Wi-Fi, then go to the settings and turn off Wi-Fi only, and that'll allow your observations to be uploaded without Wi-Fi. And if that's still a problem, then let me know and we'll do some troubleshooting. So I'm going to put my email in the chat. Feel free to reach out if you have other specific questions. Let's see. I think that's all the open questions. And then I don't know if there's anything that you guys answered from the researcher team that you wanted to highlight, but thank you for answering all those, and we'll make sure to send that out, the text from that chat in the follow-up email. Any last questions? All right, well, thank you so much, everyone, for being here. And thanks again to our research team for sharing some of the work that they're doing. We appreciate all of you that are already Nature's Notebook observers. And thank you to those of you who are just getting started now. Feel free to reach out if you have any questions or difficulty getting started. But we'll be in touch via the Quercus Quest emails, so make sure to sign up for those. Thank you so much for being here, everybody.